So RPA 8, which is about germination, I think is deceptively difficult. It seems quite straightforward, the actual practical, and it is, but there are a lot of things that can complicate matters. So let's have a look at what it is. You would have a petri dish, one of these plastic or glass dishes, and you'd put cotton wool in there, or paper towel, something that will hold moisture a little bit, and you'll put some seeds in there. So if I put 4 in there, and then you can have another dish, and what you're going to change in this is the density of the seeds, or in other words the distance between where the seeds are planted. So in this experiment, your variables, your independent variable, the thing you were going to change is the spacing or the density, and what we're going to look at is what is the effect of spacing of seeds on the dependent variable, which is the height of the seedlings when they grow. Okay, so those are our 2 variables we're going to look at. Remembering that although we don't really use this term fair test, certainly I wouldn't recommend using it on an exam. What a fair test means is only the independent variable affects the dependent variable. So only the effect of spacing affects the height. What that means in practice is we're going to have to control every other possible variable as far as possible, and we'll see in a moment that we won't necessarily be able to do that. So quickly, just to recap germination. Your seed is got a little baby plant in it really, and it's normally dormant, so it's not doing a whole lot. But when we supply it with water and a suitable temperature, which doesn't have to be that high, plants will germinate quite naturally at fairly low temperatures. You know, the beginning of spring, it's not that warm, and yet seeds are starting to germinate. So water, suitable temperature, and oxygen. Germinating seeds have quite a high energy demand, so there's plenty of aerobic respiration going on to supply that. So they do need a good supply of oxygen. So all these things are necessary for the seed to start germinating. And once they're going, the seedlings are going to be competing for these 2 things. Now, all of these we can fairly well control. I mean, you know, in a lab you're not necessarily going to control how much oxygen's present. We could do, you know, but we'd have to have some kind of sealed room or sealed box and control oxygen. So, you know, it's done reasonably. Same with, I suppose, temperature. You know, unless we're going to control everything, put it in a box and keep it all temperature controlled, as long as we're not putting one in the fridge and one on top of a radiator, we should be okay. Now, there are a couple of other things that we can't really control but are worth mentioning. So if we were using, here's a different colour, if instead of, say, we used a paper towel or cotton wool to hold the water, if we use soil or compost, which would contain, here's a useful word, minerals, things like magnesium, nitrates. Don't talk about soil containing nutrients or goodness, talk about it containing minerals. It's the correct way to say it. We can't be sure that we've got it all completely the same amount of minerals. You know, if I had a petri dish with soil in it, or a pot with soil in it, am I sure that the soil over here is the same as the soil over there? And possibly not. So that's something that, you know, is not necessarily so easy to control. Of course, you could argue with using the cotton wool that you don't get it all exactly the same thickness, and so water differences could kind of come in. But another one which is worth, I think, mentioning is genetics. So each of these seeds are going to be different unless they are clones. So if I got some cloned seeds from a plant that had been cloned, I've reduced or removed another possible variable effect. You know, the other way you could think of this, if you were doing an experiment with humans, perhaps for some medicine, you know, see how effective your medicine was. The best people to do it on would be a pair of identical twins. Well, it'd be lots of identical twins, all clones if you could, because again you've removed the genetic factor, and genetics seems to play, well, it does play quite a large role in the amount of variation between organisms. So we can't necessarily control everything, but we can try and keep, we can use the same, we could take seeds from the same plant, make it sure it's from the same species and so on. We can't necessarily control the genetics of it. Anyway, we'd let these seedlings grow, and what kind of things would they be competing for? Well, once they've started to grow, we're really looking here at light. What this little seed will do is it'll pop out some leaves from the seed, and they'll start absorbing light, they'll start photosynthesizing. At that point, the competition is on for light. Until those leaves appear, it's not going to really make much difference how much light these seedlings have. Once they've popped out, then it does. But we should control light intensity. Notice this word here. It's not the amount of light, it's not the type of light, it's intensity. That's the key word to it. Okay. And you would record the height of these seedlings. I think it's done daily on the in the practical. It suggests that you do it over the course of 7 or 8 days. Of course, you would do that at the same time each day. Again, just to remove another possible variable. Keep making sure we do it at the same time. And we could measure the height of these seedlings, making sure that we measure it the same way each time. We use the same ruler and all those kind of things. Where do you measure it from? We'll measure it from the tip of the stem to where it comes out of the leaf, for example. And we could measure other things actually. We could measure something called biomass, which you might have come across, you might not. Biomass is how much organic material, which is a posh way, I suppose, scientific way of saying how much stuff is the plant made of if you took all the water out of it. So what we'd have to do for that is we'd have to dry out our plant, get rid of all the water, which of course would kill it. So we wouldn't be able to measure biomass every day. We can maybe measure biomass at the end of the experiment. We could do that a bit more, give us some more data. And of course we repeat our readings, so we don't simply do this once, we would do repeats. Now the point of a repeat is not to make your data necessarily more accurate. Let's say I got let's say this was the days across the bottom and the height up the side in millimeters, whatever it was. Okay, and I'll just take one. Let's say I measured it and it was there. Now if I repeat my experiment, what I would hopefully get is a series of results all around the same point. And the closer they were, I would say they're more precise. So, if on day 5 I did the same thing, these results are more precise than those results because they're closer together. What that does is increases my confidence in that result, or that particular reading, my day 5 reading. Okay. Now, we also take a mean value, mean average, run out of colours. So I'd have whatever, maybe I've done 3 repeats, X, Y and Z. And remember my mean average, I would add X plus Y plus Z, and then I'd divide it by the number of readings I took, divided by 3. What this does is it tends to remove a little bit the effects of anomalous results. And that's pretty useful because I'm always going to get anomalous results. They're going to turn up. We can't control everything perfectly. These things will turn up. Maybe I've got a weird seed that grows bigger or whatever it is. Getting a mean average in here somewhere helps reduce the effects of those anomalous results. If I got something really strange that was way out, the problem is it does tend to pull your mean towards the way out result. And one of the things you can do there is you could say, well, I'll discard that anomalous result. I recognise it's there, but I think there's something wrong. And that will tend to pull your mean back a bit more to where the true value would be, or the more accurate value. Okay, even better, and some of you might have come across this, a mathematical way to do it. I'll have to put it up here. You can do something called a standard deviation, and what that basically does is it kind of chops off the top and the bottom of your results as well. So it really gets rid of those outliers. And so if you remember it, that's a useful term to have in your head when you're talking about manipulating data.